Hi, everybody. I know some of us have been chatting a little bit already, but I just wanted to say hello and welcome to GRIN Therapeutics' first engagement meeting with the GRIN community. My name is Hilary Savoie. I am the head of community engagement and communication at GRIN Therapeutics. And I'm just so excited to be in this meeting with all of you today, sort of starting out with this meeting and for the meetings to come, to kind of create a forum for all of us to share information with each other. We're really excited to talk to you about ourselves and what we're working on as a company, but we're even more excited to sort of be here with all of you and have this amazing opportunity to get to know all of you and learn from you, in recognition that we are relative newcomers to the GRIN community, and we're really eager to work with all of you in partnership, because you guys are the experts in living with GRIN disorders. So we're here to learn from you. So we have kind of an overview of just what we're going to do here today to give you a sense of the pace and what we're going to be talking about. First, we're starting with some introductions to a few key figures from GRIN Therapeutics. Everyone's going to take some time to offer a bit about their background and how they came to work in this space. We're going to discuss some of the events that led up to why GRIN Therapeutics was founded. We're going to review some of the information that we presented at the wonderful Cure GRIN conference in September. We shared a few slides there, but we want to reshare a couple of those again, just to make sure that we've covered all of that information regarding our plans to initiate the first clinical trial on Readiness and children living with GRIN 2B gain of function variants. And then we're going to kind of overview some of our upcoming points of further engagement with the GRIN community and talk about how these engagements will work together to, where we can work together to help prepare for clinical trials, as well as augment the work that's already underway in this pretty extraordinary community. And finally, we'll have Q&A with Liz Murphy-Ash, who many of you I'm sure know from GRIN 2B Foundation, who's going to offer some questions on behalf of the GRIN 2B community, but also help us moderate comments and questions from all of you live today. A few housekeeping details. Again, we are recording today's session. I just want to be sure everyone is aware of that. The goal is to be able to share that content with the community who can't attend, but we also really want to be cognizant of everyone's comfort and privacy. So if you prefer to keep your camera off, change your name, using those three dots in the upper corner, please, you're more than welcome to do so. If you have questions that you'd prefer not to ask publicly, you can chat them directly to Liz, and she can answer them without attributing them to your name. Likewise, again, you're welcome to join us on camera, ask questions through all of the methods available via Zoom. We're eager to engage however works best for you. So with that, we can move into some introductions. I think we have a wonderful set of people here today to introduce themselves and talk about how they have come to this space and doing the work that they're doing. So with that, I think I will start with our founder here, Andrea. Would you like to give us a few words about yourself and your background?
Yeah, well, first of all, thank you, Hilary and Lisa, to put all this together. I think it's a great opportunity to continue the dialogue that we started earlier in September, the Cure GRIN. And so I'm really excited to have this conversation with you all, and try to provide you all the answer to the question that we can today. But as Hilary is going to tell you, this is like a first of a kind of a series. So hopefully we'll start a dialogue today that will continue in a productive way. So, well, a few words about myself. I'm Pierre Background. I have a background in neuropharmacology. Started with some research in preclinical setting when I was finishing my medical school, and then I spent 5 years getting trained as a child neuropsychiatrist, and then went to do some academic research in human genetics, and eventually ended up starting my career in the industry at GlaxoSmithKline in 2003. And since then, what I'm doing is basically using my background in pharmacology and as a clinician to develop novel therapeutics for patients that have other neurological or psychiatric disorders. I've done in a number of organizations, and more recently with UCB Pharma, and this is why I live in Belgium, in Brussels. I'm originally Italian, but this is where I am now. And when we were doing development at UCB, we came the idea of coming up with this program that we're going to illustrate to you all. So I'll stop here and let the others introduce, and then we can get into more of the actual project and what is entailed.
Wonderful. Thank you so much, Andrea. Hans, would you introduce yourself?
Sure. Thank you, Hillary. Hi, everybody. I'm Hans Christier, and I'm the Chief Business Officer for GRIN. And I joined Pierre, Andrea, and the team in August this year, so still relatively new. And I thought I'd kind of give you two quick stories about other things that I'm involved with or have been involved with that tell a little bit about kind of my background. But I'm also a board member for KIF1A.org. And, this is a foundation that was started by my really good friends, Luke Rosen and Sally Jackson, when Susanna, their daughter, was diagnosed with KIF1A. And I think she was number 16 or 20 in the world. And, I've been working very closely with them on kind of three concepts. One is really just trying to pull together the research community. So some of these things are going to sound very familiar to what you guys are already doing, but create a forum and pull together the research community so you can elevate science. And this has turned into, we've got, I think it's monthly or every 6 weeks, you've got a research roundtable where you have up to 50 people on different continents calling in just to talk about specific topics on the science and the medical understanding. So kind of raising the awareness on the scientific medical side of things. The other was trying to attract companies to bring the best of their capabilities to bring medicines for the community. And so you've got companies like Advocacy Therapeutics, where I was before, and Ionis working in the area. And then the third thing was looking at where there are gaps in what's going on. And so we did a lot of, I don't want to get too technical, but we've done a lot of work setting up research programs and really trying to accelerate, make it easier, lower the time, money, and risk for companies to develop medicines for KIF1A. So that's one thing I'm involved with that is kind of relevant. The other is I used to work in a similar role at Advocacy Therapeutics, a company that's focused on rare neurological disorders. Sorry. And the flagship program for many years was Angelman syndrome. And for many years, we were the only company working in that area. Today, as of about 2 years ago or so, you see a few companies coming into the clinic, and there are probably about 15 programs that are approaching the clinic. So, it took some companies and efforts to come together to bring enough awareness and pull enough resources together where hopefully we'll come together and have options for the community. But through that, we also worked with caregivers. We worked with clinicians, not just from the Angelman community, but from other related syndromes, and with the FDA to try to figure out what's meaningful and how do you measure it? How do you make a difference when you're developing a medicine? So, I could go on and on. I just want to paint a little bit of a picture of some of the things I've been involved with recently. And most of my background has been in larger companies like Roche and Abbott. So a lot of that training and network has enabled me to do the kind of things that I just described here. And I started out on the science side in the 90s. So I've got a scientific background as well as a business background. But I'll stop babbling now and thank you. Looking forward to the discussion.
Thank you so much, Hans. I love hearing you talk about about the work that you've done with Luke. I think it's really inspiring, and I think it rings true with this community. So it's wonderful to hear. And Bruce, do you mind coming on and telling us a little bit about your background? Oh, Bruce, you're muted. So.
Sorry about that. Thanks,
No
Hillary, and thanks to everyone for joining and for being a part of this collaboration. My name is Bruce Lucher. I'm a board member at GRIN Therapeutics and also a physician by training, a neurologist and a psychiatrist. And during my clinical career, focused on the interface of neurology and psychiatry, where GRIN is kind of perfectly situated, i.e. pretty clear-cut neurological symptoms that patients struggle with and families struggle with, but also neuropsychiatric symptoms that are, a component part of the underlying disease process and require a lot of attention. So my clinical background resonates to this project quite a bit. I spent time away from academic medicine in various different healthcare, biotech, and finance roles. And the idea behind that work was to help me understand how all this comes together. So as you think about the work we're doing around GRIN, it incorporates science, clinical medicine, development of drugs, but it also has a lot to do with how these things get funded and how they ultimately realize maximum success for patients and families, which extends beyond science and clinical considerations and into some of the logistical and sort of reality-based thinking that biotech companies need to consider. So when I couple those two experiences, it gives me the benefit and the privilege of being able to think holistically about biotech, and in this case about GRIN. So I, away from my medical and healthcare finance experience, I sit on a few relevant boards, one of which is the Life Science Institute at University of Michigan. I'm a graduate of University of Michigan, grew up in Michigan, so I've got allegiance to the maize and blue, despite the unfortunate loss this weekend to Michigan State, tragically. But I sit on that that board, which is focused on novel development of medicines, but really early stage, discovery stage, academic type scientific work. And then I also sit on the business advisory board of the FOXG1 Research Foundation, which is an analogous patient advocacy, community engagement exercise, where a lot of the same considerations that you all are currently engaged in thinking about and that we hope to partner with you on are similar. So frame of reference that comes from a similar kind of disorder, I hope will be value added here. And then I sit on a venture philanthropy board at Cornell Medical College to get novel technologies out of academic laboratories and into companies. So all of this is to say that I try to think across the spectrum as it relates to these diseases, and have been focused for the last 25 years on getting to a point where I can materially help. And that's what Pierre, Andrea, Hans, myself, Hillary are trying to do with GRIN. So anyhow, pleasure to meet and greet everyone, and I'll turn it over back to Hillary, I guess.
Thank you so much, Bruce. Yeah, and FOXG1 is doing amazing work as well. So it's, I'm sure that many of the parents on the call, both KIF1A and FOXG1 are familiar entities, and they are doing amazing work. So to give a little bit of background on myself and how I came into this role, I have a background for the last nearly 10 years as a rare disease advocate. But I think more importantly, in this role and in this space and in most everything that I do, I am the mother of a 10-year-old little girl who'll be 11 in the new year. Her name is Esme, and she is medically, developmentally, and diagnostically incredibly complex. She, began struggling from her very first days of life, and continued to progress into, failure to thrive and epilepsy, and kind of a lot of the story that I'm sure will be familiar to many of the families on this call. And I found myself responding to that by not being able to sit still and really wanting to substantively alter the course of my daughter's life and her health and her well-being, safety, but also work in the space to help children like her. So nearly 9 years ago, I founded the Cute Syndrome Foundation, which focuses on another genetic disorder that may be familiar to some of you, which is called SCN8A, and it's often associated with developmental and epileptic encephalopathy, although there are other phenotypes. And I've worked with, the SCN8A community really just from a few years after the gene was discovered, and where we were really focused on expert clinical care and bench research, to moving to a space rather quickly and surprisingly, where we had 2 potential treatments in the pipeline for our community. And one of those I'm very happy to share is literally enrolling patient 1 as we speak, which is kind of an amazing experience to have gone through all of that. But working closely with the biotech in that space was really eye-opening for me. And I found that it was, as an advocate and as a parent, I found that working with biotech, I found partners that were sort of driven by the same things that I was driven by. Safe, fast, effective. That were, those are the keywords that we were all thinking about and working towards together. I felt very aligned with those partners. And I really feel that, felt then and feel now that it's really impossible to do this in a rare indication well, if at all, without a really close working relationship between drug developers and the communities that are those companies are trying to help. And so in my role at the acute syndrome, I really enjoyed looking for places where the SCN8A community could really prepare ourselves for clinical trials through educational options, but also prepare our partners to design the best possible trials for us, for our community as we were, and to help regulators understand the urgency in the space. And I have been thinking for a long time about ways to expand that to other communities. And so when I heard about this role at GRIN Therapeutics, it was brought to me sort of as a consideration ahead of the company even really being formed. And that told me that this was a company that was being built with patient engagement as a core principle to the company. And so I jumped at the opportunity to expand some of what I had learned as an advocate, and then and bring some of what I've learned from the SCN8A space, hopefully to the GRIN community. So that's how I landed here, and I'm so happy to be here and meeting all of you and getting to learn about your community. Thank you so much for coming and giving us this opportunity. And so I think on that note, we can dig into part of why I got so excited about working with this company, and I'll hand it off to you, Pierre Andrea, to talk about how we got here.
Yeah, no, thanks, Hilary. And as you can see, it's a really pleasure for me to have so many great collaborators, and that's going to help us in the project. So this is just to give you a little bit of a background on some key events that led to the formation of the company. As Hilary was saying, we like to be fast and speed, and I just wanted to share with you the fact that it's what we do. I mean, when I say we, in terms of the community that does science and they try to develop drugs, we try to beat timelines all the time, but none of this is very fast. We have a lot of requirements, and we're going to share with you some of the things that we need to do to be able to do clinical trials. But here, what you see here is really some of the key events that really I consider milestones. So the first one is it's actually there 2014, but I still remember the day in December 2013 when I received the preprint of this paper from Dr. Lemke, that most of you probably, all of you probably know. Johannes, published the first description of mutation on the GRIN2B gene. And, and at that time, I was at UCB with some of the colleagues, and particularly with Rafael, that discussed this, and some of you might have heard from him. So we see this publication, and we know that we have this drug called radiprotil that is a negative allosteric modulator of the NMDA. It's actually bind to NR2B. It's an inhibitor of the receptor. And we see this mutation as a gain of function. So we immediately thought that, our drug could potentially do something good on individuals carrying these mutations. But the matter of fact, there was only 3 individuals, so you cannot develop a program for only 3 individuals, although very important for those specific individuals, there's no way that we could convince UCB to do so. But the paper, and it was so important to highlight the relevance of an MDA and glutamate to infantile spasm, that is a broader, much more frequent condition. And the individuals described in this paper actually had infantile spasm. So that was enough for us to convince, for UCB to convince the company to start a program for infantile spasms. And the program was to develop everything required and necessary to do a pediatric study, because the drug was in development for another condition and it failed to show efficacy in neuropathic pain. So it was sitting there with potentially no future. So with this data, we managed to kind of resume the program toward pediatric epilepsy. We followed the field of GRIN2B, and we know that there were more mutations described, but I think there was a key event that, in addition to showing the numbers, showed the great effort the community have done to better understand the condition. And when I say the community, is the community, all of you, the parents of kids carrying this genetic variant, and the scientific community with Johannes Lemke, Tim Banke, Dr. Steve Tranellis. They all together converging this highlights, for this program was the 2019 conference where I managed to meet all the people in person, including these and many of the others that are in the call. And realized there that there was so much information that was becoming now actionable, and I'm referring to the fact that there are registry of patients, the community is exchanging information, we have the ability to describe what is a new condition. So now we have a way to say what is this disorder and what happened to patients when they have this genetic variants. And this is like critical information that I had to use to convince an investor to say, now we have a program, we could have a program that we could develop, specifically for this type of patients. And so that's what I started literally in 2019 after that conference to go back to UCB first of all to get the opportunity to have the molecule, the candidate drug, if I was capable to attract funds. And so finally, just fast forward to today, the funds became available. We assembled the company last September, and now after 2 months, we are here operating and start to make plans and everything. Well, not all the plans, but we are already working on everything that is required to make it clinical trials. And so we are here today to kind of share some of these key activities that we are doing, and just get insight also from you on how to best operationalize the program. Yeah, no, I think it's a good moment to go to the next slide. Thank you. So here a kind of very short background of what is Readiness. So as I mentioned before to you, is a molecule that we call phase 2 ready, meaning that we can start clinical studies in actual relevant patient population because we have accumulated enough information, and the dossier allow us to start a clinical study. As I mentioned to you before, the drug, the candidate drug, was in development for another indication, neuropathic pain, and under such program, there were more than 400 adults exposed to the drug. And after the first mutation described by Dr. Lemke in 2014, we started to show in vitro, to test in vitro the activity of this compound in receptor. They are coded by the genetic variants that is also present in some of the patients. And we show there that, in fact, as we anticipated, the drug, Readiness, is capable to attenuate those receptors. They are coded by DNA that carries genetic variants that eventually lead to what we describe as gain of function. So we, together with that, we also come up with a program that preclinical testing of the molecule in a variety of preclinical models. And we are continuing to do so in more specific models. Now, GRIN-related models are becoming available. So we show that the compound has activity toward the seizures. And, and as part of the initial idea to go and target and study a patient with infantile spasm, we managed to start a trial where in infantile spasm patients, in infants, between the age of 4 and 11 months. And we tested the radiprudil actually for up to a month in these 3 infants. And the radiprudil in reality showed that the drug is safe and tolerated and managed to control the spasm, the seizures that were present in infants. And in one patient, became seizure-free after testing radiprudil, while the other had a dramatic reduction of their spasm seizures. So that's kind of all the background that allow us to start now a new program that will be directed to individuals, to kids that carry gain-of-function mutation in the GRIN2B gene. So that's kind of, in a nutshell, the background of what we have in our hands and what it will allow us to do the clinical study as soon as we can, but not immediately, because we need still to put in place a number of activities that I guess you will see in some of the next slides.
Maybe I can take us through a couple of high-level points.
That'd be great.
So, our intention is to build off what Pierre was just talking about. We just put a couple of very high-level work streams here, and I think as we go through, our intention is to make sure that the different activities are clear, so that you understand kind of the things that we need to do in order to bring the medicine forward, potential medicine forward. Obviously on the bottom with the 7 to 10 months, we have to prepare a package of information that shows the FDA and EMA in Europe that what we have is safe and it's made appropriately, and we've got a rationale and a reason that it makes sense to bring this forward. Everybody wants that. That's what Hillary talked about in the beginning. So there's a whole process of summarizing all of the information and sharing with the FDA, not just about why it's safe and compelling, but also what are we going to measure and how are we going to measure it, and what does a specific protocol look like. So that takes some time to pull together. In parallel, we'll also be looking at, we talk here about the pediatric formulation of radiprudil, so making sure we've got a medicine in a form that is easy to take for the kids, right? But there's a whole work stream behind that as well, where just the manufacturing process, so that it's made to spec, it's safe and it's made the way it's supposed to be. We know what we're giving is what we've set out to make. So that's another work stream that takes. We're estimating timelines here, and we'll keep you informed on how those things go. And the top box is kind of what we started, I think, at the Cure GRIN conference, and just want to make sure we've got an open dialogue as we go forward with this. We really run it in parallel, and making sure that you understand what we're trying to accomplish, and we understand what's meaningful for you, and we have an open dialogue about what that means. There are scientific elements that Pierre and Hillary talked about earlier on, understanding what it means with the variants, and then what does that mean from your experience on a daily basis, and working with you to understand those things, working with the physicians and the scientists, and ultimately being able to talk through that, not just with this community, but also with the regulatory authorities. So that's an ongoing dialogue. And I think today we just wanted to frame that we have these different work streams coming together, and let's set up a cadence in a way that we can continue the discussion and share with you how we're going, and make sure we're learning from each other. But those are kind of the key points that I wanted to highlight. But Pierre, Andrea, Hillary, Bruce, did I forget something that I was supposed to say?
No, I think you hit everything. And I think what's so important here, and I can actually move into the next slide, I think, is what you're highlighting, this ongoing conversation is so important. And we are so fortunate that already there is, as Pierre Andrea highlighted, there's already this incredible background work that's been done by your advocacy groups in this space. GRIN 2B Foundation, certainly Cure GRIN and GRIN 2B Europe have been just doing amazing work. And it's so important. And in fact, I joke often that one of the first things that I did when this job came to across my desk, I guess, as a potential, was I went on Facebook. I didn't go and look at what was happening in scientific papers. I went on Facebook to see what was happening in your community, what parents and caregivers in this community were doing, because of so much of what Hans and Pierre Andrea were highlighting is so important, that we need to know what's meaningful to measure, so that from you, so that we can do a good job of getting through these next hurdles together as a community. And just to highlight some places where in the SCN 8A community, we were able to provide that kind of feedback through kind of an organized advocacy effort, was in providing feedback for formulations. We did a formulation survey and we're able to provide really substantive feedback on formulation needs and preferences. We've been able to help identify clinical trial sites, and which you can see in Liz's map here. She's already done the beginning work of, these are the patients, here's where they are. We're in touch with patients in these places, and helping look toward centers really for where the patients are. And then also advocacy groups doing the amazing contextualization of what it means to participate in various efforts. Like, for instance, the registry, which I think we can, I know we've talked a bit about the registry earlier today and the importance of the registry. And this just is an incredible resource that your community also already has, the GRIN portal and the registry, that helps, first of all, as Pierre Andrea highlighted, understand the path to getting a functional analysis done of any variant that hasn't already been analyzed, which is incredibly important. It's also a really useful resource for the community to communicate with regulators, information for clinical trial design and enrollment, and helping everyone in the ecosystem coordinate as we try to bring medicines forward. I think it's also really meaningful on a daily basis to help improve clinical care and allow all of you to make better treatment choices for your children as we move forward. Is there anything else that you'd like to add to that, Pierre Andrea or Hans or Bruce, about the registry?
No, I think it's hopefully will come later on the discussion, open discussion on the relevance and how important or useful this tool can be for us and, well, in general, the community.
Yeah. Okay. So I'm going to just move to this to just quickly talk about some of the further engagements that we have planned, and then we can move into the Q&A. I want to be sure that we have plenty of time to answer questions from all of you. So obviously today, November 1st, this is really just, a meet and greet and trying to get to know each other and meet each other where we are. In a week, we are offering a really incredible workshop with this amazing advocate in the rare disease space, whose name is Rachel Callender. And she's going to be giving a workshop on, really, it's on caregiver expertise and kind of turning the language that we use to talk about our children, that we're also used to hearing deficit language with respect to our children, what the piles of papers that we have that say what they're not doing, and really turning that on its head and starting to talk about what our children are already awesome at doing. And you can see this is my daughter Esme's awesomeness form. And we made these in the 8A community as well. And it was just a really wonderful exercise to remind all of us as caregivers what we already know. And parents use these as resources, for their educators, for their doctors. But it's also really important, I think, for professionals in this space, clinicians, researchers, drug developers, to understand these sorts of details about our children, because although, yes, of course, we are looking to address deficits and potential treatments, we're also looking to make what's already extraordinary about our children able to be more extraordinary on a daily basis and more easily accessible for them. And that's really what's at the heart of this. So I am super excited about this workshop. I know Liz is as well. And so we'll have Rachel next week. And then a week after that, we're going to have a discussion really about, about clinical trials. And in fact, a lot of the content will be designed based on the conversations we have today and next week, to make sure that we're bringing to you the information that you need to understand about what clinical trials are and, what we need to be learning from you. And then throughout 2021 and 2022, we're going to continue to engage with you. We have some resources that we're developing, but we're also looking to you to help us understand what your needs are, what you need to support your lives, and how we can have the most impact. And I think it's really important just to highlight again some of what Hans mentioned, is that we really need involvement from the community, because we need to understand what's important to you as caregivers, what's important to your children. So we understand how GRIN disorders manifest in your daily lives and what would make a difference in your daily lives. And if we can understand what matters, we have a much better chance of actually measuring that in a clinical trial, and bringing forward a medicine that is able to help address and known to help address those issues. And again, I just want to highlight some of what Pierre Andrea said and Hans said, that the clinical trials are incredibly complex by their nature. And we really want to make sure that we're working closely with you to make sure that the trials that we design are as user-friendly as possible, and that we're thinking proactively about how to build the right trial for this community. So with that, I think we can, Liz, if you'd like to join us, turn on your mic.
Mm-hmm.
Thanks so much for being here today.
Thanks for having me. Hi, everybody. Okay, so there was already a question in the chat that was answered. Thank you, Pierre Andrea. But I think I have a bunch of questions, and if anyone else has questions, again, feel free to throw them in the chat or chat me directly. But I will get started with my questions. And I do think this question that was asked here in the chat about the GRIN portal registry, and if that's a requirement, I want to discuss that. Okay, so my question is, can families enroll in this clinical trial without also being enrolled in the GRIN registry? Or perhaps a different way to word it is, will you be enrolling people? Will you be contacting, working with Dr. Benke and Dr. Lemke to find the GRIN2B gain of function patients? Or will there be some sort of enrollment portal on your website that anyone can just find on their own, or through us and Cure GRIN promoting this?
So yeah, Lisa, it's important question. So there are several elements. So first of all, I want to make sure that we kind of give it the right expectation on what we're doing there, because I want to make sure that, although we would love to have as many kids involved, that we have constraints that are the constraints dictated by, in any clinical study, when you start, you need to start gradually, and we cannot start enrolling every patient that can be enrolled and it will be imposed even by regulators. So it's not even our choice. We have no choice. So we have to gradually, expand the number of patients. And so just to make sure that, the number is not going to be enormous, so it's not going to cover all the eligible population. We're trying to make our best effort to see how we can compress timelines to include as many patients, but there will be a limited number of kids that we can enroll. So then the question: how are we going to identify the kids that they are more suited to be enrolled in a trial that could benefit the most? Primarily, our first point of contact for reason of expertise and knowledge is, of course, Dr. Benke and Dr. Lemke. They have the registry. We can go through the list of without knowing who are the individuals, just to make sure that this is all going to be anonymized. But we have our kind of criteria that we can share with you. Now they're not final, but at least we have a sense of what are those criteria. So we need to see who are the patients that have those criteria, and in terms of genetic genotypings or clinical variants or symptoms, and then decide where best to start, because that's also the key question. Now, if this is going to be the only way we're going to do, no, we're going to do it in a way that makes sense. I mean, talking to you directly through all the families that we have here in this group, a variety of methods. The importance of the trial, of the portal, as there are several relevant points about the portal. One is that we can look at the data in aggregate and better understand the disorder. So the more patients are there, the better information we get out of the portal. And also when we do the selection of the patients, we can also understand better how can best operationalize the study. So basically, it's a container that gives a lot of information for us. It is very useful. But as I said, it's not the only one. The key requisite, one of the key requisites in terms of criteria, is really understanding the genetic variants and the consequences of the genetic variants. So we have some information out there in terms of the literature or out of the portal, but it's not going to be exhaustive and in final information. We would like to expand that information to make sure that we are as precise as we can be. Precise meaning in terms of understanding of the biological consequences of the genetic variants. So I guess, Liz, I gave you maybe more points that maybe you asked for, but I know that there are some of the points that are quite important to me. And I don't know, Hilary or Hans and Bruce, anything, of course, you guys want to add to what I said, or any other questions, clarification?
Yeah, I think the important piece just for all of the families is to understand that this is an incredibly important resource for the community, for everybody who is in the ecosystem, including drug developers, but not only drug developers, but that as we look to enrolling, and as Pierre Andrea was careful to say, this will be initially a very small trial. We can't enroll everybody. And this is a resource to help us in identifying patients, but it is not the only resource.
And also to make it even more specific on the relevance, as I was saying, we would like to corroborate the information that is out there in terms of, what Dr. Steve Fernandez has done, right, in functional consequences. We will work with him and the entire community to say, okay, this is the information we have, how we can expand it. But the information we have, the repository of the information is the portal. This is where we know which variant has been assessed and which one has not been assessed. So what we're going to do is going to start there as a kind of baseline, as a starting point to say, okay, now we want to expand the information. So that's also to give it another relevance of the portal.
And then maybe just to complement what Pierre Andrea and Hilary said, we'll go into more detail around how clinical trials are set up and how they're run another day, but just to address your question of how do you get into the trial, we will have a few sites, call it, for the US trial, we'll have a few sites in the US, and those are picked by understanding what are the most convenient sites. There are a number of factors that go into those, right? And then, I think it's a normal process for speaking with your physician, looking to see if you meet the inclusion criteria, and all of those kind of things that then you would be brought to the trial. But these are things that we'll go through it in a subsequent meeting. I just wanted to make sure that we did check that part of that part A of the question off as well.
Thank you, Hans. Liz, do you want to move on to another question, or shall we, I'll let you decide how to.
Yeah, thank you all for answering. Okay, let's see. There was a question in the chat that I'm going to ask. Why are you starting trial efforts with the GRIN2B gain of function variant specifically?
Yeah, and this is like because of the mechanism of action of what of the molecule. What does the molecule? And unfortunately, can only address gain of function, at least for what we know. Because the gain of function means that the protein, the receptor, become overactive, and the drug is actually an inhibitor of that protein, of that receptor. So it's the idea is that we compensate this overactivity with the molecule, the binding to the protein, inhibit that overactivity. So that's why we're focusing on gain of function.
Okay, thank you. That makes sense. So I think you guys were saying that we'll talk more about the criteria for enrolling in future webinars, or can I move on and ask questions about that?
Yeah, no, you can ask it. Absolutely. It's just that
Yeah, so I understand you don't obviously know at this point the entire criteria that one would need to enroll in this, but at a base level, we know it's going to be gain of function variant, and I believe, correct me if I'm wrong, the individual needs to have uncontrolled seizures or just some sort of seizure activity.
Yeah, it's an important point. I remember we discussed it early days, a couple of years ago, and we are very mindful of the fact that it's not only seizure, and some of the kids, they don't have seizure at all, right? But they have all sorts of other problematics. So it's so there is two aspects to answer this question. So why are we focusing on seizure? Well, first is because this is like quantifiable and measurable, and we have to quantify and measure the effect of the drug if we want to progress further in development. We need to be able to say to the agency, to everybody, that there is a benefit. And so the best way to do that, if we can do in an objective way. And seizures, as you unfortunately know very well, they're very objective effect that they can be counted, and there is a lot of experience in doing trials for seizure. So that's one of the reasons why we put a kind of emphasis there. And of course, we realize that seizures are also problematic, as you all know. The other aspect is like, how are we going to do with patients that have no seizures? The short answer is that we don't have a clear-cut plan at the moment. We know that we would like to address that because it's important for you and for the kids. And we know also that the drug is likely to have an effect in a lot of other symptomatology, because we are impacting on the primary reason that is causing the disease, right? So we might have an effect on behavior that are problematic and all other sort of symptoms. So at the moment, as I said, we haven't finalized our plans, but we would like to consider, seriously how we can do that to include the non-seizure patients.
Okay.
Yeah, that's a core part of why we need to work together, is to reemphasize what Pierre Andrea said. As you are setting up a trial, you need to show what's quantifiable, what's reliable, and what's different. And then within that, you've got a range of things you can measure. And the ones that are standard and used very frequently are the seizure-related, what we call endpoints, or things that you measure. And then there are a whole bunch of other things that we call scales and different things we can measure that some are more robust and understood by the community and the FDA, and others are less so. But even those that are reasonably well known and understood, we need to make sure that they are used, measured, interpreted in a way that's meaningful for the situation here. And that really takes a lot of dialogue and back and forth. And it's actually a plus that we have We're not coming here and saying we know the answer. We'll work with you to get there.
Okay, thank you guys. So I guess piggybacking on that, assuming the criteria does mean, you know, seizures, active seizures, which I understand fully why you would need that. So you need something to measure. So my daughter has a gain of function variant and does not have seizures. So, you know, I understand she most likely wouldn't be able to participate in the trial. But so is it correct to say that if the trial is successful in the patients that do have seizures, this could be something that the ones that don't have seizures could eventually use down the road?
Yeah, I think it's not that. I mean, we could start earlier than that. I mean, it's not that we wanted the trial to be, because it could be successful or unsuccessful on seizure, but could have an effect on behavior, or I see neurostorm as you know, very problematic, a variety of other things. So the idea that I was discussing, that we are still discussing, is how we could do, can we include a subset of patients that don't have seizure, and try to understand from that how we can include a broader population in the next study. Because the thing that we are doing in the first studies, why also we are so cautiously moving into this, because we need to understand where the drug can have an effect and where we can maximize its value. So if we do what we do in the first study, is really to learn what we could apply in a subsequent study. Now the question is, should we include the patients or kids that don't have a seizure immediately in this first study as a subset? Should we include it as a separate study, a trial? All this question, we don't have there are no easy answer to them. And what we would like to do is to really explore also with the FDA, with the agency, and get their advice, how we can best position this. Because it's as I said, you know, it's not that we're doing something like business as usual. There's nothing like this in this type of development or trials. So that's where we are.
Okay, thank you so much. And I know we're running to the hour, but I see there's a bunch of questions in the chat. So let me go through these. Okay, let's see where we are. Thank you, Hillary, for answering the question about the registry. Okay, here is a question. I'm assuming this clinical trial will also aid in determining the effectiveness to all GRIN gain of function variants? Or I'll just reword it. Will this trial aid in determining the effectiveness to all GRIN gain of function variants? Or do you just think this will specifically be for GRIN 2B for now?
So we have data for 2B at the moment. We have legitimate beliefs that it should work also in the other gain of function variant, particularly GRIN 1, because of what I hear from Steve. Well, because of what I know of the biology, but Steve Tranellis has tested in some, not radiculate, but other pharmacology, and he suggested that it might have an effect there. Potentially that could be true for GRIN 2A. The point is that it could have an effect there, but before we commit to this, we need to produce data. And that's what we are in the plan to do, produce data for GRIN 1, GRIN 2A, and I would say potentially GRIN 2D, that's all to be understood, to be tested and studied. And
I think if I can add to that, Pierre Andrea, I think what's really important for the families to understand about this process is that it's a stepwise process, right? We're doing experiments in a very particular way, and then we're gathering information, and then if there is information that suggests taking the next step, then we take that next step. So the idea is to move forward carefully and based on where the science drives us, but the hope is to broaden. I think you can hear from what Pierre Andrea is saying, that the hope is to broaden the patients that we reach as much as humanly possible, as long as the science and the regulatory bodies are behind behind doing so. Is that a fair assessment, Pierre Andrea?
Great, thank you. Okay, so I think this next question, I think you already talked about this a little bit, but about what other symptoms could be possibly alleviated. Neurostorms, cognitive development, and motor skills, communication. So I guess the question is, yeah, do you expect that any of those symptoms could be reduced?
The potential is all there. Then there are questions on, can we reliably measure them, or can we expect an effect within the time of the trial? And so the trial will have a, the testing for a number of weeks, could be 8 or 12 weeks, depending. And then, we'll have to make an assessment on what's happening, because we need to understand if it makes sense to continue. So then if a patient that will have a benefit, they will be able to continue in what we call open label extension. So they will roll in another study and will continue to do longer observation. And the reason I'm saying this is because in reality, all the symptoms can be affected, as I was saying before, but some of them might take more time to show an effect. So we will have to learn, basically, potentially there, everything, but, we will have to see.
Okay, great, thank you. Okay, and then the last question I see here is, what are some things that parents can do?
Sorry,
Yeah.
to break up, parents can do?
Can do
What
now.
are some things parents can do now?
Oh, can be done now. Okay, well, I would. My first reaction would be like, help us and understand how can we best do this. Asking the good question that you're asking now, and when we go more in the specific of the trial, this will be critical information on what we should measure, what we should pay attention, and tell us when we show you the outline of the study to tell us, look, have no limit of what you will say to us. Say this is a nonsense. We're not going to be able to cope with this. Don't do it that way. Really be open and tell us we're here to try to do the best of the job. So we need your help on this.
Absolutely. And I think actionably, at this moment, if you are not already enrolled in the registry and are willing and able to do so, that link is in the chat. And I know Liz and Keith and other advocates in the space are sharing that information. You can be sure to make sure that gets shared again on the heels of this meeting if you're interested in enrolling. And then we will have these other opportunities up as soon as next week and then the week after as well, to engage with us and continue to give us feedback and help us understand your community and the things that you want us to know about you, so that we can get to work on designing this trial and then, as Pierre Andrea said, show you what we have and hopefully continue to get more feedback from you. And I think it is really important to underscore what Pierre Andrea said, that we are willing to hear whatever it is that you feel we need to know about this space, and we really want to learn.
Okay. I see one final question in the chat. Is there any use of genetic editing using CRISPR in this approach?
In our approach, no, there's no. I don't know if it was related to our approach. Our approach is a small molecule. We call small because just the chemical structure of a small size that is like any other simple pill that you take as aspirin. So there's nothing different in terms of chemical. The only difference is that, of course, it binds specifically to this receptor and does what I was describing before. So there is no other activity that we don't impact on any DNA or RNA or anything like this.
Thank you. That's it. And I think all my questions got more or less answered through all the discussion.
I would say if there are more questions, don't wait until the next session, You have Hillary's phone number, call anytime or any one of us. So, keep the back and forth going.
Yeah, people are welcome to send questions to me too, and I can pass them on.
Yeah, and Liz, I see one question I can address very quickly that we didn't talk on the chat. There was a question about the medication. When the kid does get in a trial, does the other medication get stopped? No, not at all. They're kept as stable as possible to avoid the confounding effect. But if something happened, they can be modified. So in essence, that's what the way we do studies.
Fantastic. Well, I think perhaps on that note, we can close the meeting. I know we're a little over time, which is wonderful because it means we have lots of questions to answer and things to talk about. So I think in closing, I just want to say thank you to everyone from the GRIN community for showing up and asking your questions, sharing your comments, and we look forward to engaging with you here. And thank you so much, Liz, for coming on and moderating and doing such a lovely job of communicating the questions from the community to us. Please, everybody, you are welcome to join us and this time next week. Although I think in Europe, because of the slight difference in daylight savings time, it's actually an hour later next week for you guys. So please join, and we'll have a really wonderful workshop on with Rachel. And in the meantime, if you have further questions, pass them to Liz, pass them to Keith, pass them to Sandra. And I put my email in the chat. You're welcome to email me directly. And with that, I think we can close and say thank you all for being here today.
Thank you, guys.
Thank you.
Thank you, everybody.